Hello everyone, in this video I'll be explaining on the topic of negative staining. This negative staining is a staining technique which is used to study the morphological shape, size and arrangement of the bacterial cells that is too difficult to stain. Example like the spirilla. It can also be used to stain cells that are too delicate to be heat fixed. It is also used to prepare biological samples for electron microscopy. It is used to view bacteria, bacterial flagella, biological membrane structures, and proteins or protein aggregates. These, which all have a low electron scattering power in this technique. The background is stained, leaving the actual specimen untouched and thus visible. Coming to the principle of this negative staining requires an acidic dye such as India ink or nigrosin. India ink or nigrosin is an acidic stain. This means that the stain readily gives up a hydrogen ion, proton, and the chromophore of the dye becomes negatively charged. Since the surface of most bacterial cells is negatively charged, the cell surface repels the stain. The glass of the slide will stain, but the bacterial cell will not. The bacteria will show up as clear spot against a dark background. The reagents required are India ink or nigrosin stain. Coming to the procedure here, place a very few small drop more than a loopful, less than a free-falling drop from the dropper of nigrosin near one end of a well-cleaned and flamed slide. Remove a small amount of the culture from the slant with an inoculating loop which is sterile and then disperse it in the drop of stain without spreading the drop. Use another clean glass slide to spread the drop of stain containing the organism using the following technique which is mentioned here in the diagram on the screen. Rest one end of the clean glass slide on the center of the slide with the stain. Tilt the clean slide towards the drop forming an acute angle and draw that slide towards the drop until it touches the drop and causes it to spread along the edge of the spreader slide. Maintain a small acute angle between the slide. Push the spreader slide towards the clean end of the slide. Being stained, dragging the drop behind the spreader slide and producing a broad, even thin smear. Then allow the smear to dry without heating and then focus a thin area under oil immersion and observe the unstained cells by the gray stained. So when observing under the microscope, place one drop of the oil immersion on the smear when the smear is fully dried and then observe under the microscope. So what you observe here is you can see the bacteria is seen against a dark background. Here in negative staining, the background gets stained while the bacteria or the microorganisms remain unstained and can be well seen against a dark background, as you can see here. So that is the explanation of negative staining, which is very useful to stain those bacterial cells which are not stained by ordinary stain or which are too thin or too small to be seen under a microscope, or some structures of the bacteria like the capsules can be seen by this method of negative staining, and then the capsule can be observed under the microscope with the help of this negative staining.